Good morning, everyone. I am very happy to share part of our work addressing biodiversity shortfall in meiofauna with you. And this work is presented by me and Gustavo, but Diego Fontaneto also collaborate with us in several ideas in our first paper published 2 years ago. So I am bringing these results, and as well I am bringing an update. We are bringing an update of ideas and how we are progressing on this topic. So what we want to propose here is about when we think about biodiversity shortfalls, we have to remember and also promote the idea that more than a scientific jargon or a complicated term to try to give a theoretical importance to our research, we need to remember that is a current responsibility with our understanding of biodiversity. I want to bring a idea, as the famous David Attenborough say in this new testimony on Alive on Our Planet. This talking about biodiversity is not more about saving our planet anymore, it's about saving ourselves. Thinking about that, we think meiofauna biodiversity can tell a very inspiring history to promote several reflections. And because that, I will start this history, bring the animal that inspired my scientific career for over 15 years. The history started very simple. Once upon a time, there is a worm on its grain of sand. So when we start studying meiofauna or how animal life in the earth, and we try to understand how diversity relates evolutionarily and how species are distributed on our planet Earth, we notice that our question is not so complex at first moment. They are very simple. And then in themselves are our shortfalls, as we will see along these presentations. Questions like who are they, how many are they, what are they doing, who are they distributed, how and when did they get there, and how do they interact. They seem quite simple at first, but we have to ask, why is it so easy to answer this question? Of course, there are some processes that we must consider related to this spatial and temporal scales of variation when we think in a macro scale view. At this point, we cannot leave aside our limitation as human and researchers. As when we think about plate tectonics in a global evolutionary scale, or even when we think about tide is high, but I'm holding one, and we can ask ourselves what happened and what we can mentionally sampling and understand. And when we understand our limitations, we notice this real mass of a process and patterns. And as we find it humanly impossible to understand everything in an integrated way, in a linear way, sometimes we can call them complex system. So, well, thinking about that, the proposal to address the biodiversity of our meiofauna is an invitation for us to try to clean up this mess and direct our theories, concepts, and methodologies. The question here is how sitting in our microscopes can we integrate taxonomy, phylogeny, food webs, and oceanography process. So I think that's why we think the global shortfalls, this is a great tool proposal by Hortal and collaborators that we should consider better in our meiofauna classes and our research lines. Here, I try to use this laser pointing. Here we bring in an example of a simple exercise we can do together with three groups, what they call it nematodes, copepods, ginatostomulids, and annelids, just the interstitial ones. For example, if you get the linear shortfalls, which deals with our limitation or ignorance to know all the species names, we can say that we still have a lot of work to be done to entitle meiofauna. We have a lot of species to describe. For distribution, talking about Wallacean shortfall, I think that many species of nematodes and copepods have already been well studied, but more work needs to be done to address more precisely. But for ginatostomulids and annelids, we still have a lot of work. When you think about Prestonian shortfalls, species abundance, I dare say we know a bit about abundance of several nematodes and copepod species. I'm not sure about ginatostomulids and annelids. We still have a good way ahead. And then we can follow this exercise. For example, if the works of Wolfgang Steres, Rice, Ferris, you know that ginatostomulids occur in fine to coarse sand near to redox layer. Then you can address the shortfalls for Hudsonian, the environmental requirements. As well, you know something for nematodes, and thanks to many ecological works of several colleagues, they are here. And likewise, for the evolutionary history of some annelid groups, since Westerheid, Puschka, Joanne, Thorsten, and of course, the team that I belongs with, led by Catherine Vorso, Alejandro Martinez, we have advanced in evolutionary understanding of several families. We still have to work on it, but we know something. I think this is a nice exercise to do, to think about what we should do to understand how we are describing and understand the biodiversity of meiofauna. And why not how our lives depend on it. Although this is a very attractive philosophical subject, I want to come back to a point that I think is essential to reflect on our methodologies, the dimensions of our work and our frustrations, which for us is one of our real limitations to address the biodiversity shortfalls for meiofauna in an integrative way. When we think, and this is in the textbooks of meiobenthos sampling methodologies, we separate our chapter themes like quantitative sampling or qualitative sampling. While quantitative sampling is strongly used for ecological estimative of abundance and distribution, qualitative sampling ends up being more used for taxonomy, functional morphology, and evolutionary history. First, we note the difference in the sample volume dimensions. To count we take a very small quarter, like this one, for the identification purpose. Many times, we take a single individual from an entire bucket. Then, following the approach, we have an inversion of routines. To count is better to fix before and identify after. To describe specimens and other things, first we sorting and morphotyping them, and so we identifying, and depending on the purpose of the study, we fix then with the best fixative for each proposal. Of course, you must be thinking now here, maybe the big difference I am talking about is hardy bodied and soft bodied meiofauna. Yes, I think so too, but beside that, both methods can generate frustration during the discussion of our results. We, as you can see, for the quantitative sample, sometimes we miss data and about evolution, functional morphology, and traits. And when we are talking about evolution and morphological and functional morphology, we miss data about abundance and distribution. So you can see they are often complementary for each other, no? So what we can do then is the question that you make several times, like, is there a fixative for everything? Is there a sampling strategy for everything? I think it's very difficult, no? But look at back and in the past as Giard, maybe 1904, or Swedmark, 1964. We can also see the works of Rimani and all their students and Eveline Marcos. And you can see in these papers, they did a beautiful works integrating abundance and the abundance of species, the distribution, species description, great adaptative, providing great adaptative and evolutionary insights and the environmental requirements. I agree with you, times have changed and changed a lot. Nowadays, if you take a simple Google Scholar search with the term meiofauna, since 2016, it returned 7,000 findings. If you use meiofauna in distribution, you will find 5,000 findings. Meiofauna in taxonomy, 4,000 findings. Meiofauna in evolution, 4,000 findings. I believe this shows us how much we work and dedicate ourselves to describe the biodiversity of meiofauna. So what we're trying to say that we are addressing very enthusiastic this biodiversity and the shortfalls. We are not so many, but we are very enthusiastic. So how do we improve what we're doing then? What methods can you get to study meiofauna and address the shortfalls? And again, is there a single method to address these topics integrated? What we're proposing here, and then Gustavo will talk after that, we can think in this large amount of data for morphology, the large amount for molecular data, the larger amount of presence and abundance data. And we have to think how they are connected. So we have to think cross disciplines, and each topic can integrate our data from each other. And in particular, we have to sharing our data to each other. Thus eliminating much of your initial frustration and limitation. And what should be a great motivation for us is to keep going and believe that the meiofauna can inspire our society to take care of our planet health. So I will finish this here, and I will let Gustavo continues how we are addressing the shortfalls now.
Okay, thank you, Maikon, and thank you, Michaela, for the presentation of ourselves. I will start here sharing. Yes, sharing. Let's see. Oops. Is it working now? Can you see the presentation? Is it yes? Yes, okay. Okay, continuing with the rationale that started by Maikon, the question that we should do, I think, or I did myself, is how does each of us look the meiofauna? In the first moment, of course, we imagine these beautiful animals. In some minds, I think this may pop up, but in most of our brains, this something like that will show up. So some of us think about meiofauna as A, T, G, C, others as morphological characters, and sure, the ecologists like myself are more into ecology. These quantitative tables, numbers, followed by environmental matrix as well. And how do we merge all this knowledge to address the shortfalls? So I think this is the question that Maikon started in this methodological perspective. It's one. I saw already in the chat, okay, what about eDNA would be a promise? Of course, it will be a promise to merge different shortfalls. And at the end, we are going to end up, if we start to merge this information, we end up with many spreadsheets. So we have two families of spreadsheets, basically. We have this sampling spreadsheet where we have this site identification with our coordinates. Then we have the species list, which we found in these sites, all the metadata associated to it, the environmental data, pollution data, methodological data. Then we have the other family of spreadsheets, which is this related to the species, which is the species ID, species name, at best when we have it. Then you can add up with morphological characters, traits, phylogeny, stable isotopes, and anything that belongs to the species, not necessarily to the site. So, but the thing and the difficult starts, okay, this is beautiful, all these spreadsheets, you just need to combine and start to analyzing. But when we merge data sets, so I will merge my study with the study from Maikon or the study from Jeroen, it ends up that we have something like that. So many gaps. Now, and the difficulty is, okay, what do we do now that we don't have this information, the exact information across all the sites or the exact information across all the species? So then they arise the next question is, how do we deal with the lack of information and how do we fill the gaps? And I see that we have two strategic or two ways to fill the gaps. One, I call it the strategic sampling. So we move toward this special site that we don't know nothing about, these very deep Mariana trenches. We go there and we sample our meiofauna from there. We can think about the location, we can think about the taxonomies, they do that a lot. So I need this missing puzzle between two set of families, which is the evolutionary path of this character. So we can do that as well. But there is also another strategy, and I think all of us do that a lot with our on meiofauna. Is my precious sample. But I think we have this inferential approach that we should explore more and more and more, because we do have, the planet is big, the amount of species is enormous, and the amount of spreadsheets that we have to fulfill are even larger. So this inferential approach, we should start to give more attention. And how do we do that? What is the logic of the inferential approach? Okay, given that we have I put this small square here just to give as example. So given that we have this set of morphological characters which are related to these traits, and I know the phylogenetic position of some of these species, can I infer the phylogenetic position of these other species that I don't know anything about the molecular data, for instance? How can I place there somewhere in the evolutionary tree? We can play around with these squares, around this, all these tables, and trying to infer where the information is missing. What is my guess? So what do I guess to find in this certain place? Or what do I guess to find in this species, let's say, functional trait? No? And we do that a lot as well. But I think we should put more attention on that. And here I bring 3 recent studies on, because when we do inferential approach, when we choose to do that, we need to have clear assumptions. And to have clear assumptions is what we, as meiobenthologists, what do we believe? What will happen if here now in Guarujá, where I'm here now, sitting now, comes an oil disaster? So what do I expect? Okay, and these 3 studies, recent studies, I think they try to make an effort to organize our minds on that. The first one is the need of hypothesis-driven designs and conceptual models in impact assessment studies. So do we need, what is at the community level here? The study deals with individual level, population level, community level, but here I just bring 2 graphs on community level, where species richness, biomass, abundance in response to, for instance, contamination. Now, what is going on? What will happen with these 3 indexes? The same we try to do based on the community structure, on the MEDS plot. So what do I expect if contamination happens? What do I expect, or disturbance happens? What do I expect that will happen with my MEDS plot? So basically, the study is bringing this to our mind. The second study is the motive of this presentation, which is the addressing biodiversity shortfalls. The conceptual idea, the assumptions that are doing is how do we integrate the patterns, the ecological patterns that we see now, that we are investigating, which are the processes that are shaping these patterns. So, but these processes, they have to be seen from the ecological and evolutionary perspective, because we have a set of species that are there, living there, based in ecological, environmental restriction, but also evolutionary restriction. So this paper try to bring us ideas of how to merge them in a theoretical way. And more recently, I found this paper from Michaela and Sommerfield, where they also bring a conceptual model on how meiofauna will respond to different disturbance and how is the cascading effects. So all these 3 studies are doing, they are organizing our thoughts, our finds in conceptual models, so trying to stimulate, so that we can test these hypotheses, whether they are truly what we are saying or they are completely wrong. And actually, what we are doing in this exercise is we want to transpose the knowledge across the space, our knowledge that we already have in the literature, across environmental conditions, across the time, and across the biological dimension. So we are filling the gaps by transposing this knowledge. And here I coming back here for these strategies of strategic sampling and inferential approach. I will show you 3 studies just to stimulate your reading so that you can see these papers, although maybe they didn't cite the shortfall itself, but they certainly contribute to address the shortfalls, is this recent published for the response of life history traits of estuarine nematodes to a pollutant. And in this study, the idea is to move beyond the traditional functional traits. Actually, the authors discuss that within the same trait, you can have completely different response according to the life history of the nematodes, whether it is a, I don't know if all of you remember the R strategies, the K strategies, where investing in your children or investing more in your somatic growth. Then the second one, it's from Curini Galetti, the contribution of soft-bodied meiofauna taxa to Italian marine biodiversity is a huge step, I think, because as Maikon said, the soft-bodied taxa, we have very little information about the distribution range because of the fixatives that we use. So this information comes from the taxonomical studies, and they compile a lot of studies to start to have an idea on the distribution range. And then in the inferential approach, on the other hand, here I bring to you this study from Danilo and myself, where we use the simulation approach. So the title of the article is Simulation-Based Framework to Explore the Importance of Non-Selection and Selection Processing in Structuring Ecological Communities. And basically what we do is to take our the theoretical approach. So what do we have from theory? You simulate our guess, what we think that will happen. We simulate in the computer. We generate tables of communities. And then at the end, we see whether our simulated table match our observed table. So this is the inferential approach. We are trying to fill the gaps. So if this method of simulation works, we can fill the gaps of our missing information. And how they contribute. So just to make a link from what Maikon said, the life history paper contributes certainly for the biological traits. So for this shortfall here, that is barely pronounceable, Rocurian shortfall. Then we have the paper from Galetti contributing for sure for the Wallacean shortfall, the distribution of species. And we here in this simulation paper, we are using the data of the environmental requirements of the species, also the biological traits, to simulate our communities and see whether we can get the real community, the observed community. So we are contributing with the expectations from the Hutchinsian and how current shortfalls. And if you want to continue now in filling these gaps of the tables, I think one of the most important aspects is collaborative work. Alone, it won't be possible like to move rapidly forward all these tables, in filling out these tables. And here I want to show you all the initiatives, some of the initiatives, not all, some of the important initiatives that we have in our meiofauna community, which is the Meio School, the Scalidofra workshop that happened recently. The Meiolive is also a collaborative work so that we can share our knowledge before even being published. And I think we need even more to explore the data sharing approach. If not completely open, but maybe among us, the meiobenthologists, we can share our spreadsheets and see how they can be connected. So this was my part of the talk. I want to thank you, everybody, for watching. I want to thank Michaela for the Jeroen, Florian, and Giovanni for the opportunity. If you would like to contact us, feel free to send us an email, and we are open for questions, discussions, as you want. So thank you very much. I will stop to share now.